Good morning to everybody. I'm going to talk about the genetic diversity of white potato relatives from Ecuador. As a little introduction, a cultivated potato is this one that you can find at the supermarkets, like here, for example, the Mona Lisa, and is well known as Solanum tuberosum. The cultivated potatoes have high yield, uniformity. You can find, like, for example, the potatoes are with the same size, shape, and color, but most of the cultivated potatoes are susceptible to some disease, and for that reason, they are pesticide dependent. On the other hand, the white potato relatives they have low yield, they have variable size of shapes and colors, as you can see in the picture. Like, for example, you can find dark, pink, a lot of colors and shapes of the potatoes, and also they are very small. But the most important characteristic of the white potato relatives is that they have a big source of genes with disease and also to resistant to abiotic stresses like heat, drought, and freezing. For that reason, the scientists use this type of white potato relatives to create new varieties, and they are used in potato breeding programs. So, my project was in Ecuador. We evaluate the white potato relatives from Ecuador. Ecuador is in South America, is this small country that you can see, but we have a high biodiversity. So every project, every research that we do in order to know more about our biodiversity is very important. We use these 8 wild species that you can see: Solanum macaule, Solanum albicans, albornozi, andreanum, chiliacense, Chomatophilum colombianum, and minutifoliolum. Also, I want to say that the cultivated potato is Solanum tuberosum, but these are the wild potatoes. So the number that you can see is the number of accessions that we use. One accession is 1 point that the scientists collected throughout Ecuador. Like, for example, Solanum albicans has 4. One will be here, another here, here, and here. So are specific points that were collected. In total, we collected 91 accessions, and we did a molecular characterizations. We use microsatellites. Microsatellites are small DNA sequences that are repeated consequently throughout the genome that is no coding. So in other words, we have 3 accessions, for example here, or plant 1, 2, and 3. And these are like keywords, like, for example, agate. And if this keyword repeats different times, like, for example, he's 7, in the 2 we repeat 4, and this 12, this is something that we are looking for, because we say that a microsatellite is polymorphic and give us information to say that one plant is different from the other. And we did this. First, we did a microsatellite screening, and we were looking for information in some papers that were reported that were very informative. We select 50 microsatellites, and we did a lot of tests. So we were looking that they have a good amplification with a PCR. They are polymorphic. We say that they are polymorphic. You can see here there are little points, but they have different weights. So if they are polymorphic, they have to be different, and we can see the difference between them. And finally, we optimize the PCR. Like, for example, here you have a picture of the polyacrylamide gel. So in this group, we have 1 microsatellite, and below we have another. Each line is 1 accession. So here there are the 91 accessions, and we put a number. Like, for example, the weight will be here 200, this will be 202. So we have the data for all the microsatellites, and finally, we have the results. So we know that the microsatellites that we analyzed were highly informative, And we have this dendrogram. So the dendrogram was, we have 10 groups, but there were 3 main groups. The first one that is at the top, we have this in black, and it was the cultivated potatoes with Solanum macaulay. In a lot of papers, we know that they are very close, so here we also confirm that. And the second group is this one, and the third group is this one. We saw that there was a mixture of all the species that you can see here. And this is very important because we elucidate that they are derived from process of domestication or also from hybridation or gene flow. So this is something that we are evaluating. And this is important because we can use this difference that the species have in order to do a potato breeding program and create new varieties. Thank you.